Hi everyone, I thought I'd help you out with some of the genetics problems that you're going to have to do and hand in this semester. Let's start out with a typical autosomal recessive problem. Here you have the condition of oculocutaneous albinism, which is inherited in an autosomal recessive pattern. This means, of course, that both copies of a gene in each cell will have mutations if one has the disorder. You will have to have both alleles as recessive to have this disorder. Most often, the parents of an individual with autosomal recessive conditions each carry one copy of the mutated gene, and they don't show the signs and symptoms of the condition themselves, and this is called the carrier state. If a carrier for albinism will be heterozygous for this disorder, a big A, little a, and they mate with someone who is recessive for the disorder, meaning that they have the disorder, little a, the likelihood of their children also inheriting the condition is far greater. So in this problem, you need to demonstrate the likelihood of having the disorder for any child that is born to a mother who is a carrier, big A, little a, and a father who has albinism. His genotype will be little a. So you always should start out your problems with writing down exactly what you know and what you need to find out. So you know in this case, Dad has albinism, a recessive condition, so his genotype is little a. And the mom is a carrier, so she's a heterozygote, but she doesn't have the phenotype of having the disorder. So she is big A, little a. And what we're going to do is find out the probability that when these two mate, that they will have a child who has albinism. So the child must have the genotype little a. This is how you set up your Punnett square. We'll go to the answer on the next page, but I hope you sit down first and pause and write out how you're going to make this Punnett square on your own. So we set up our Punnett square for an autosomal recessive cross between a heterozygote and a homozygote. And what we have here is the mom's genes across the top, a big A and a little a. So her gametes or her eggs can either be an A or a little a. And then to the left we have Dad's gametes or his sperm, which will always be little a. So one little clue you can do here is I have it set up where it shows both of his potential gametes, but since he always produces little a, you could just drop off the bottom set of boxes and know that 100% of the time he's going to have sperm that is little a. So here we have all of our combinations of possible zygotes that are formed in the boxes. A big A, little a, or a little a. So if one parent is a carrier and the other has a recessive disorder, their children will either have the genotypes big A, or little a, exactly like the parents. So you have 50% chance of being a healthy carrier. You have a 50% chance of having this recessive disorder, and there is a 0% chance that you will be a healthy non-carrier. Let's move on to an autosomal dominant condition. And the one that we talked about in the textbook was Marfan's disorder. Okay, and we're just going to do it here as a purely autosomal dominant disorder. We're not going to talk about all the other aspects of Marfan's disorder that we talked about. Okay, so Marfan syndrome is caused by a dominant allele. So inheriting just one copy of a dominant allele will cause the disorder. People who are heterozygous, big A, little a, have the disorder, just like the homozygous, who are also dominant on both of their genes, big A. What are the chances that a child will inherit Marfan syndrome if a heterozygous male mates with a female who does not have the syndrome. So once again, write out what you know. You know that dad is big A, little a. He's a heterozygote for the disorder. And mom does not have it. So she's little a, little a. Here the normal condition is recessive. And we need to find out the probability that a child is going to have Marfan syndrome, either big A, little a, or big A, big A. So work out your Punnett square and join me on the next slide. Up in the right-hand corner, we see our pedigree chart for the autosomal dominant condition, Marfan syndrome. You can see that the affected persons are the father in the parental generation, and in the F1 generation, or the first filial generation, where there are 4 children, half the children have the disorder and half do not. Marfan's is autosomal dominant, so all you need is one allele to have this condition. Across the top of our Punnett square, we wrote the genotypes for the eggs, the maternal gamete, for the paternal gametes, the sperm, big A and little a. And then to the left-hand side, we wrote the genotypes for the gametes for the unaffected mother, little a and little a. And again, just as with the previous Punnett square, since mom can only give a little a, you really could have dropped off the whole bottom row there and just had 100% little a in one box. Okay, I hope you can see that. All right, so we work out our genotypes, and once again, the possibility for the children is either children can be big A, little a, or little a, little a. Those are our 2 genotype options, 50-50, right? 1-to-1. So if a parent has a single copy of a dominant allele, their children will have a 50% chance of inheriting the disorder. And that's what we see up also in the pedigree chart. 50% of the children inherit the disorder. And 50% of the children have a chance of being entirely normal and being completely recessive. Little a, little a. We're now going to get you started with doing one of the problems from your homework assignment, problem number 1, exercise 8 from page 116 of your lab manual. Here you're going to have multiple different crosses between different colors of tomatoes. We will do the first one to get you started. In tomatoes, the red fruit color will be signified by the dominant allele, capital R. It is dominant over the yellow fruit color, which is represented by lowercase r. The first cross they have assigned you looks very familiar to the last crosses we've done, which is the heterozygous, big R, little r, crossed with a homozygous, recessive, little r, little r. So I think you already know how to do this, but let's do the cross on the following slide. And here we've crossed the homozygous recessive, little r, across the top. And we have the gametes that are possible to be formed by our heterozygous red tomato plant, big R and little r, to the left-hand side in the red boxes. And the possible zygotes or plants that we come out with are either big R, or little r, little r. So we have, once again, a 50-50 R, big R to little r to little r ratio. Big R to little r, little r. And our phenotypes will also be 50-50. Half of the plants will be red and half will be yellow. Okay, now let's move on to a different type of problem. This is a sickle cell problem, which is problem number 7 from exercise 8 on page 117. Here Normal red blood cells are due to a dominant gene, which will be represented by capital letter B, while sickle cell anemia is represented by the recessive allele, little b. In this problem, a carrier of sickle cell is considered to have normal red blood cells. In reality, it's a little bit more like incomplete dominance, with a sickle cell trait being a possibility for the heterozygote condition, but that's just an aside. Okay, so a normal man whose father had sickle cell anemia marries a woman with the disease. Both of the woman's parents were normal, and the man and the woman have 3 children, 2 that have a normal phenotype and 1 with sickle cell. We need to list all of the family's genotypes. As always, we start out our problems with listing what we know, so we kind of have the map of what we can figure out. Okay, now for Dad, what we learned was that his grandfather, the paternal grandfather, had sickle cell disease. So therefore we know that the paternal grandfather had to be little b, and could only give little b gametes to any of his children. Therefore, our father must be a heterozygote. He must be big B, little b. Okay, and our paternal grandmother can either be big B, little b, or big B, big B. He had to get a normal gene from mom, but we don't know what her other gene is. Okay, now our mother, we know, also has sickle cell disease, so her genotype must be completely homozygous recessive, little b. And maternal grandfather and grandmother are normal, but each of them had to give the mother a little b gene. She had to inherit from both of them. So therefore we know that the maternal grandparents are both heterozygous, big B, little b. Do you understand that? Okay, so now we have Dad, we have Mom, we have the paternal grandparents and the maternal grandparents. And then we need to figure out the possibility for the children. So we're going to determine a cross between Dad, who is a heterozygote, and Mom, who is a homozygous recessive. And this looks like a super familiar cross because it seems to be all we're doing today. So this is how you set up the cross between Mom and Dad. Mom can only donate gametes that have the little b allele. Down the left-hand side in the red boxes, we have Dad's possible gametes. He can have sperm that have the capital B allele or sperm that have the lowercase b. In the white boxes, we see the possibilities for all the zygotes that could be formed. And that could be big B and little b, or little b and little b. So once again, we have our genotypes being 1-to-1, or 50-50, or 1/2 and 1/2, whichever way you choose to recognize that. Genotype, 1/2 of all the kids that are born will be big B and little b. And 1/2 will be little b and little b. Now, just as an aside here, let's remind ourselves, you know, these 2 parents could have all babies that are little b. Statistically, over the long haul, you know, if they had 20 children, they should turn out with approximately 10 being one genotype, the big B, little b, and 10 being the little b. Now, most of us don't have 20 children. And every child is a new event. What we're saying is that with each birth of each new baby, that baby has a 50% chance of being one or the other genotype. Then when the second baby comes, that baby will have a 50-50 chance of being one or the other genotype. Okay, it doesn't mean that if baby 1 was big B, little b, that baby 2 must then be little b, little b. It's the 50% chance for each new event. I hope you understand that. Okay, and so the phenotypes possible will be half will have a normal phenotype but will be a carrier, and half will have sickle cell disease. Okay, you will have several problems that are a little bit more complicated where you'll have to examine 2 traits. Okay, so now we'll do a 2-trait problem. And remember to always first set up your problem with each parental type. For the following problem, we're going to look at cows, and solid color patterning will be dominant over spotted coloring pattern. And we'll have the fat body type being dominant over lean. So now we're going to mate a heterozygous solid pattern cow who is fat, and we're going to mate that cow with a spotted lean cow. Now try to set your problem up, and then we'll set up the Punnett square. Okay, so what we know, solid is dominant over spotted, fat is dominant over lean, and we're going to have a doubly heterozygous solid fat cow with a spotted lean. So first, let's write the genotype for our doubly heterozygous solid fat cow. So that cow is capital S, lowercase s, capital F, lowercase f. Okay? The S and the F, the capital letters, are dominant for solid and fat. And we know they're heterozygous, so they have the recessive form of the other genes. And we're going to mate that with a spotted lean cow. So if the cow is spotted and lean, we know that cow is completely homozygous recessive. SS, both lowercase and both lowercase f. Okay, so we know then that the heterozygous solid fat cow can produce the following gametes. Can be capital S and capital F. It can be capital S with a small f. It can be small s with a capital F and small s with a small f. And our spotted lean cow that's completely homozygous recessive can only produce little s, little f gametes. So on the next slide, I'm going to set up this box in a different way, just to show you what you can do and what you can drop off. So this time I put in our Punnett square all the 4 possible gametes that the solid fat cow can produce across the top. And since our spotted lean cow only produced one type of gamete, I just put 1 box across the bottom. And it still works out with the correct ratios. And you can do that if you only have 1 type of gamete. Okay? So it makes it a little bit easier to go through the problem. So our first one can get capital S and capital F from our solid fat cow. And let's say it's our bull. If our bull is only lowercase s and f, then they get the lowercase s and f from the bull, right? And so we end up with our doubly heterozygous cow in that first box. Then in our second box from our solid fat cow, we inherit the gamete that is a capital S and a lowercase f. and it mates with our all lowercase bull. So we end up with a solid capital S, small s, and a small f, resulting calf. Okay, so it would be solid and lean, right? Then we have our cow that produced gamete that is a lowercase s and a capital F, mating with our same bull, and that produces a zygote, which is both lowercase s's and a capital F and lowercase f. So we have a spotted and a fat cow. And then our last zygote is produced by inheriting a lowercase s and lowercase f from our solid fat cow and inheriting a similar gamete from our bull. So lowercase s's and lowercase f's. So this one looks just like its daddy and is all recessive, so it is spotted and lean. And if we do all of our genotypes, they're 1 to 1 to 1 to 1, right? Okay, you have a 25% chance or 1/4% or 1/4 chance of getting each different genotype. And we have 4 different resulting phenotypes. You will also have a number of blood type paternity questions. So let's go through one example here. Let's say Mariah has blood type A, and in court she claims that Nick is the father of her baby, Tommy, and Tommy is born with blood type O. Nick is blood type B. Is it possible that Nick is the father of Tommy? So first you have to set up what you know. So Mariah is blood type A, so she can be either IAIA or IAIO. Nick is blood type B, so he can be either IBIB or he can be IBIO. Tommy, the baby, is blood type O, so he's totally recessive and can only have IO alleles. So let's work it out if both Mariah and Nick are IAIO and IBIO, if they're both heterozygous. So if that were the case, I put Mariah's genes going across the top and Nick's possible genotypes for his sperm going across the bottom. So if Mariah's gamete carries the IA gene and Nick's carries the IB gene, it will result in an IA-IB zygote or child with blood type then AB. If Mariah's gamete is type O, IO, right there, and that unites with a gamete from Nick that was IB, then the resultant baby would be IBIO genotype, which would be type B blood. If Mariah donates a gamete that is IA and Nick donates an IO for his gamete, the zygote that forms from that would be IAIO, which would be type A blood. And if Mariah donates IO in her gamete and Nick also donates IO in his gamete, the zygote that unites to form that is IOIO, type O blood. So you see every possible blood type can arise from this mating. So yes, it would be possible if they were both heterozygotes. Now, let's do a problem if Nick is not a heterozygote, if he is IB-IB. Here we have the mating in which Nick is homozygous for the IB gene. Andy mates with Mariah, who is a heterozygous for type A blood, so she is IA-IO. And you should know that if I say Mariah is heterozygous for type A blood, that the only other allele that she could have would be the IO allele, right? Okay, so what we see here is that with Mariah donating gametes that can be either IA or IO across the top row in blue, and Nick can only donate one type of gamete, the type B, okay? And that is in the blue down the left-hand column. Now we could do this another way if I want to set it up, and let's look at that. So on this slide, I have the same thing where Mariah is IA and IO across the top, and I only have one row because Nick can only donate one type of gamete. So 100% of his gametes will be IB, so we just only really need to write in that one row. And we can see that the resulting mating from Nick donating only IB and Mariah donating either IA or IO as a gamete or as an egg forms only two options. You will either have a child who is IA-IB or blood type A, B, or Nick donating IB and Mariah donating IO, and therefore their child would be IB-IO, which would result in a blood type B. So no, if Nick was homozygous for blood type B, then Tommy could not be his son. Okay, let's look at color blindness. Color blindness is sex-linked. So if you haven't moved on to the second genetics chapter, you might not know how to do this. But hopefully you can learn how to do this and understand from here. So in sex-linked genes, the allele is carried on the X chromosome. So the father, who is XY, or any male child, only gets one dose of an X chromosome. So if you have a diseased X chromosome, that all sons, all males, will be affected. So let's look at a normal phenotype mother who is a carrier for the color blindness gene, and she mates with a man who has normal vision. What are the chances that they will have a colorblind son? Okay, so let's set up what we know. We write that color blindness is recessive, carried on the X chromosome. Mom is a carrier, and dad has normal vision. So now figure out what those two genotypes would look like. Write it down, start to set up your Punnett square, and then move on and check to see if you did this correctly. Here, mom carries the recessive gene for color blindness on one of her X chromosomes, whereas dad, who has normal vision and only has one X chromosome, must have only a normal gene. So mom, therefore, can donate either the normal gene, X with a capital C, or mom can donate in a gamete a defective gene, the X with the little red C in lowercase. Okay? Down the left-hand side of our Punnett square, we see that dad can donate the X that has the normal gene on it, or dad can donate a Y chromosome. And from these different gametes, we'll now figure out what the different zygotes or resulting children can be from this mating. Let's look at the first row, in which dad is donating X chromosomes. So a daughter is what will result from this mating. Dad donates a normal X chromosome in either case, and in the first case, box 1, mom also donates a normal chromosome. So you have a totally phenotypically normal female child with no color blindness. In the next box, Dad donated his normal X chromosome, but Mom donated the defective chromosome. So what you have there is a carrier female. She has one normal chromosome that makes her not be colorblind, and she has one abnormal chromosome that gives her carrier status. On the bottom row, Dad is donating Y chromosomes, so he only has sons. Okay? Here we see that the first box of the bottom row of the possible zygotes, Dad donates the Y chromosome and Mom donates her normal X chromosome. So therefore you have a normal non-colorblind son. And in that last box, we see that Dad donated his Y chromosome, but Mom donated the X chromosome that has the colorblindness allele on it. And the son does not have a dosing of a good colorblind gene from Dad, so therefore that son ends up with color blindness. So what we see here is that 50% of the sons will be colorblind, or 1/4 of all children if we consider both sexes. And this is where you can remind yourself of how this turns out. We have one affected son possibility, we have a carrier daughter possibility, and then we have a completely normal son or daughter as a possibility. Now, one way in which these questions can be tricky is in paying attention to how the question is asked. Do they ask what percentage of all the children will be colorblind, in which only 1/4 of all the possibilities of children is colorblind? Or will they more likely ask you what percentage of the sons will be born colorblind, in which case you choose 50%. Do you see that? You only have two options for son. One will be the Y chromosome with the normal X, and one will be the Y chromosome with the abnormal X chromosome inherited from Mom. So 50% of the sons, 25% of all children, in which case you're considering both sexes. There's another way that this question can be tricky, so let's look at that too. This is when we want to consider X-inactivation, or what you've learned as being called the bar body, and considering this with sex-linked conditions. So in this previous example, in which we had the normal phenotype mother who was a carrier for the color blindness gene, mating with a normal man, is there any way that the daughter could end up colorblind? So we already know what this mating looks like. We know that Mom carries one abnormal gene, which we see down here in red for the carrier status. And we know that Dad is normal vision, so he has a normal X chromosome with the capital C that's in black. So let's look at this for what happens with X inactivation. All right, so we have our mating in which our carrier mom can either donate the normal chromosome, the X with the normal allele for the color blindness gene, or Mom can donate an X chromosome that has the abnormal gene on it. We're talking about daughters right now, so we can really completely ignore the whole bottom row where a Y chromosome was inherited. And Dad can only donate to his daughters the normal X chromosome. So looking at our two daughters, we have one daughter that has completely normal chromosomes, and then we have the other daughter that looks like Mom, and she's a carrier status, right? However, can that carrier ever end up being colorblind? And yes, indeed, this could happen. Remember, when you have a bar body formation, one of the X chromosomes is randomly inactivated in every cell in your body, and another X chromosome is good. And it's completely random which one it is. So overall, you're going to have, 50% that inactivate the bad chromosome in one cell and 50% that inactivate in another cell. But in some people, it might be 90%, right? So maybe in one female that's born, all by randomness, 90% or 99% of her good chromosome is inactivated, and it's always her bad with the color blindness gene that's the active one. Well, in that instance, you could end up with a colorblind daughter.